So my name is Brian Caron. I'm the Director of Operations and Development specifically to NeuroHub, and as Dave mentioned by JB, a co-lead with him specific to the NeuroHub project. What I'm just going to introduce you to today is just a very quick, I would say, backgrounder for those of you who may not have seen some of our prior training, just about what NeuroHub and the ecosystem is, specifically that, and then we'll jump right in specifically to talk about the UK Biobank and the LORIS Data Query Tool. At that point, I'll hand it off. We'll hand it off to two people, actually, beyond myself. So I'll then introduce Samir Das, who specifically is responsible for the team looking after the LORIS DQT. And then we'll hand that over to Xavier, who will guide you through some exercises, and then we can get some practical hands-on experience in using the DQT. So the outline of the workshop for today is as follows. Like I said, I'll give you some background context along together with Samir with regard to NeuroHub, LORIS, and UK Biobank. And then we're going to go through various exercises that will help you understand about the query, the data that's being queried in the UK Biobank, ways of, and then how to get access to that in terms of disseminating that and pushing that out to other systems where you can run some processing on it, specifically within CBrain. And we'll summarize that with some features. And at the very end, we'll have a link to a short survey to get your feedback on things. So just a few things into logistics to work through, since we do have a large number of participants, and even though we've been, as of tomorrow, been working on doing this mode of living, if you will, for about a year, just a few things to remind people about working in Zoom. So just to facilitate, people being connected from various home connections that have limited bandwidth, I just ask that you kind of, you can turn your camera off when you can. We're happy to see it. It's actually, we encourage you to keep it on because in the sense that it actually helps us give us some visual cues and visual feedback, but at least more from a speaking point of view, unless you're actually say something, please try and mute, just that way it keeps the noise level down. And if you're connected, if you can change your participant name, just so that we know relative to our list of registered participants, your name, so we can address you accordingly. As you should have acknowledged, the workshop's being recorded. And so we're going to make it available on the website within probably about a week to 2 weeks after the session here, so that you can go back and look at it. And also we can then point it to any of your colleagues for people who couldn't make it, so that they can observe what was going on. But really, as much as this is you'll see a mixture of a demo and a mixture of hands-on exercise, we really encourage you to work on the exercises to really maximize the benefit that you get out of it. Given, of course, that we know that some people may not have access to UK Biobank by various processes that are needed to be in place. In those cases, that's something you can follow up through afterwards with the exercises once you are registered. Oops, go back to the window so I can change slides properly again. Again, if there's any questions, though, we understand that we can't, very quickly survey the room and look for waving hands or confused looks if people have any questions. So we just ask that if you do have a question, you can indicate one by typing in the chat window they have a question, or even just type the question directly. And then the presenter will, either address it or one of our helpers. We should have a number of helpers around the room. If you look in the participants list, you will see those people identified with the kind of postfix tag of helper beside their names. And one of those people, either on the LORIS side, or the UKB, the Biobank side, or the CBrain processing side, will be able to address your question in that regard. We also have some breakout rooms that will be present there just in case we have to get into some more screen sharing, but we shouldn't need to do that probably. But if you do need help, if you're really you're stuck somewhere, things like that, you asked your question, or you really have some trouble that you need some dedicated help, raise your hand in the Zoom room. Like I said, we have people looking for your helper request, and one of the people will be able to assist you. And then also one thing, because it is a workshop based upon exercises, you'll see various points when Xavier is guiding you through the exercises. There'll be various checkpoints where we're going to say, okay, have people achieved certain milestones or completed certain tasks? And, of course, in a room, you can raise your hand. Here, we're going to ask you to kind of do the same thing. You can actually put a yes or a no if you're having some issues. Just that way we can kind of survey the room accordingly to get an idea where people are at. And then we can then correspondingly address any particular issues that somebody may be having. So that's just to help us work within, like I said, this Zoom context. Are there any questions about the logistics pieces? Okay, so just moving on, and I'll try and be brief in this regard. So hopefully everyone would have taken a look as part of when you register, the prerequisites in terms of some of the links that we shared about some of the prior training sessions where we gave some background about the fundamentals of what NeuroHub is, and some of the other specific workshops. But really, NeuroHub is an ecosystem. It's a platform providing integration of different various platform tools and focusing and centering around CBrain and Loris as core components. So we have a very specific web-based user interface that you'll get very experienced with today. And then leveraging that underneath the hood, there's things like an API layer that you'll maximize and take advantage of to show the relationship and the interconnectedness between Loris and CBrain. There's the data query tool, which is why everybody's here today to see on how to use that, and particularly the integration with a major tool, from a major data set like the UK Biobank is a prime thing that we're advancing as part of the overall platform. So you'll see this through the web interface. You'll see basically the conceptual ideas of a web interface, which provides you a portal of entry. You'll find ways of working with data sets. You'll do that interactively for the data query tool, even though we have other interactive data exploration interfaces that we won't talk about today, but we're part of a prior workshop. And then you'll make use of what's called the processing factory, which is organized and orchestrated on the back end by CBrain. So you'll get to exercise essentially all four of these conceptual spaces today. So one of the main parts, and just to save time, I'll skip over the prior piece, the prior slide. So one of the big things you're going to be getting to work with today is the UK Biobank. And as I mentioned, some of you have already accessed through the allocation that specifically is tied to NeuroHub, and some of you may not. But just for background information, so the entire UK Biobank sample, so right now that we host within NeuroHub through the agreement, represents about 200 terabytes of data. That includes not only just the imaging, which I think is the predominant use case for people attending today, but also more genetic information. And we also, as part of that, take care of things like the versioning. We also take care of participant removal that's announced by the UKB. And the idea behind this allocation, it's a widespread scope application so that we can consolidate into one installation this large data set. This is not a data set where you want multiple copies sitting around. You want to be able to have a singular copy where we can organize it, but then you can also have multiple accesses to it. So that direct access we provide through the NeuroHub, the portal, but on the back end as well, we provide access to people who may use Compute Canada resources such as Beluga and other systems. And of course, through Loris, which is the prime location where the data is kind of stored on the front end. So with that, not only do we have the main data sets, but we also have derived data. We're processing this data continuously through various pipelines and putting back into the system these derived data samples. In general terms, you're going to say, okay, what is really in the UK Biobank? I won't go through the full list, but other than to say we have a documentation page on our wiki that shows in very detailed view what those elements are that are available within the UK Biobank. It includes not only the tabular data that's there from various formats, whether it's CSV or R even, but of course covers all the imaging modes, which my colleagues here will be able to speak more directly towards, but also in the genetics if there's groups that are interested in that as well. So that we're continuing to add in terms of the exome data as well. But if there are any samples, specifically or data fields or return data sets that you're interested in, through our support line, you can send us an email and give us some specifications about what you're looking to have added. Again, these details are in the link, which you can find from our NeuroHub documentation page right off the portal. And then we can work with you in that regard. So that provides a lot of detail there about what is within NeuroHub, both via the portal, but also through the interface we enable through Compute Canada. And my last remark in general about the UK Biobank, and this ties, like I said, to the number of people attending today. If there's I would say, administrative process to go through because it is not an open, it's not a public data set. There is a registration process that people have to go through. So you have to register both with the UK Biobank, but also through NeuroHub so as to have that access coordinated. So these are the two, I would say, processes that you have to go through. But once you go through that, though, you get access to both through the web-based platform as well as Compute Canada. And again, we have on our documentation pages a step-by-step, blow-by-blow process listing for how to go through that. So at this stage, I think if there's no questions, or even maybe there's more specific questions that people may have, I have to reopen the chat window into a different section here. But I'll hand it over to my colleague, Samir Das, who's Associate Director, specifically looking after the Loris side and the Loris elements within the lab and looking after the UK Biobank. So I'll hand it over to Samir and he'll be able to talk more about some background about Loris and where that fits within the context and its capabilities and so on. And then we'll continue on with the rest of the workshop. So Samir.
Okay, thanks a lot, Brian. So basically what I'm going to do is first I want to introduce a couple of people that are key to this mission. So first and foremost, Xavier, who has worked quite extensively on this particular UK Biobank instance. He's going to be the one taking you through the workshop, showing you how everything works. Feel free to ask questions at any time in the chat, and there'll be times even afterwards where we're going to make sure that everyone goes through and understands very clearly what's being done. There's other people that are kind of supporting today, but maybe that's a bit of a misnomer because some of the people that are supporting are actually people who are senior developers that have spent many years working on exactly this infrastructure. So one of them is Dave McFarlane, excuse me. And he actually was the person who created the entire data querying tool that we've created. It's on its third version in its lifetime. It started in around 2004. It was a MySQL database. Since then, it has moved to something called CouchDB, which is what's known as a NoSQL database. Dave actually developed this entire system from scratch. And so, you know, his contributions are quite extensive here. And so he probably can answer all sorts of questions that are more on the systems level. Other people that have been working on this quite a bit, and they're here to support with any specific questions that you might have. Rita, he is also a senior developer in the Loris team. He's, there he is. He has actually implemented this kind of technology for something that is related to our cause here, which is the MNI's Open Science Repository. So just recently we have, you know, exposed all of that data to at least registered users and internally at the MNI. Soon there is an open layer coming there. And so he's done a fair bit of work there as well. So he's here to answer any questions, especially like more user support questions on how people are interacting with the system. And finally, Alize, as well as another Loris developer who has spent a fair bit of time improving the front end. Now, so there's always improvements to this technology. It's quite elaborate, and I'll just spend a couple of minutes explaining a little bit about the technology itself. So first and foremost, Loris is a data management platform. It's been around for about 20 years, and we've systematically developed this system to kind of mimic the workflows of what neuroimagers or neuroscientists are typically doing. One piece of this is the data querying tool. There's a whole bunch of functionality that Loris has aside from the querying aspect. What we found is that researchers are particularly intrigued with, you know, how you can query multimodal data. And so this layer, so to speak, is here to basically help with trying to bridge all of these very large data sets together. Now, what we're about to show you is kind of taking this to the next level. We have had many instances of Loris, the querying tool in Loris, working over the years for projects on the scale of several hundred or even a few thousand subjects with all the different kinds of modalities on the imaging side or behavioral measures or demographic information, and even some summary genomic data in recent times. And as well, we've included all kinds of biospecimen samples as well, and that's a relatively recent feature within the Loris environment. But what ends up happening is researchers love this capability because you can make very elaborate queries. You can create these queries without being a programmer, and you can filter down onto the exact subcohort of a data set that you would want in terms of your analysis. There's other bells and whistles that are attached to this, which I will show you, or Xavier will actually take you through all of this in a bit. But the point is that for something like the UK Biobank, which is an extremely large, it's an order of magnitude, just for the imaging data, it's an order of magnitude larger than what we're used to dealing with. So we've had to do all kinds of interesting scaling. We've had to, there's a lot of work that went into this to make this work in such a way that was like efficient and optimized and useful to, you know, people like yourselves who are looking to kind of mine this data. So in a nutshell, what you're about to see today is a very elaborate construction that does not really exist in too many places. If you have an extremely large data set like the UK Biobank, which is 500,000 subjects, you have to be very systematic in how you get the cohort of data that you're specifically looking for. It often requires you to be someone that's fairly technically versed, even just writing scripts to parse down and extract what you want. At the very least, you have to have a decent understanding in statistics or statistical tools to be able to get to that point. And so our goal here is to make this easier, right? So it was quite a challenge to take something of this size and be able to query in a cross-modal way quickly. But this is our first rendition of that, and it's hopefully you find it fruitful and useful. So that'll be the proof. And there are, I'm sure, many functionalities and features that we could still add to this. And that's where we're happy to hear what you think is important as after this workshop or during this workshop. If you have any thoughts about what you'd like to see, what might be more efficient, we're all ears. And I guess with that, I will probably leave it there in terms of the high level. So why don't we actually take a look and see what we've created, and hopefully make sure that everyone is familiar with it. Is there, what's the next slide, Brian? Right. So, there's a few overarching points here. There's a lot of functionality, whether it's in Loris, whether it's for data collection or APIs, and there's a demo database. So if you're curious about the greater aspects of it, you can take a look there. But I think now is a good time to maybe call Xavier up to the podium, so to speak, and he can take you through all of this. Any comments that I might have missed at this point?
I think it's all good. Thank you, Samir, Brian.
Yeah, I'll stop sharing my screen. So if you want to then take control of that.
I'll do that.
So just one last thing before Xavier gets going. So what you're about to see today is you're going to see the Loris querying tool in action. You're also going to see functionality that extends beyond Loris, which is the ability to move data from in an API-centric way to CBrain, for example, or NeuroHub, which is an extension of CBrain, and be able to then run, whatever kind of tools on it. We will have very specific examples so that they can be done in a timely fashion. As you can imagine, mining the amount of data that we have here, you could run queries that could take minutes to hours, and you could run image processing algorithms that also could take days or even weeks to run. So we're not going to do that necessarily in this particular workshop. We have specific features that are much more attainable, so that everyone understands what functionalities exist. But you're free to play, and with this, now that you should all have accounts at this point to do what you're supposed to do. Like Brian said at the beginning, there's two groups for this workshop today. There's the UK Biobank allocation that falls under a specific allocation for which people actually have permission to play with. And then there's external visitors that may have a UK Biobank access under a different allocation. This just shows you all the possibilities of what you could do with this data that are make it hopefully much easier for you to process and analyze the data. So with that, I will now turn it over to Xavier, and feel free to take us through it.
Yeah, and I'll just quickly mention while Xavier's about to get started, there are a number of other projects that we know that indeed have UK Biobank data, for example, on the genomics side. So we're even discussing with the Genome Center, for example, for in those cases, because we want to obviously minimize the number of copies or any duplicates that may exist on platform systems such as Compute Canada. So we're already in conversation with them about, let's say, coordinating the accesses so that we at least are good system citizens in that regard. Sorry, go ahead, Xavier.
Okay, so hi everyone. Today I've prepared a couple of exercises, but first I'll explain what we did for the ukbb.loris.ca. It's a Loris instance that is housing all the imaging data of the UK Biobank dataset. It's roughly 40,000 participants. And along the images, we've added all the tabular data for these participants. The tabular data had to be organized. The next slide is about data organization, but roughly it's 182 instruments, and there's about 20 terabytes of nifty images that are available through the DQT. As for duplication concerns, the images are not on the Loris host. They are kept in the Beluga on Compute Canada, and they are fixed in SquashFS files that we are mounting using the SSHFS that are spawning singularity images. So as for organization, the typical Loris schema have one or many projects in which there's a couple sub-projects, and for each of them, participants or candidates are added. Each candidate has visits, and for those visits, there's instrument containing variables. For the UK Biobank, there are instances with instances index. There's participants, and for those participants, there are data fields that are organized in categories. It's effectively a tree of categories. To add them to the Loris database, we've translated that to having data fields, main category, the leaf categories of the tree, being instrument, and each instrument contain all its data field. I'll briefly go to the UK Biobank showcase page. If you want to use the UK Biobank, I think this is the Bible. This is something that you should get familiar to. It contains a lot of information, but I'll stick with the category trees. So this is a tree of category, population characteristics, in which we can find baseline characteristics. And in this leaf category, there's effectively 6 data fields. So in the Loris instance, we can expect to have a baseline characteristic instrument with all those variables. I'll go back to presentation. So in the data query tool, as mentioned by Samir earlier, this is a powerful tool that allows, without having to write any SQL code, to select a set of variables and apply a filter of a population of participant sessions and extract the relevant variables. It's harnessing the power of NoSQL database. We are building pre-built query, and once the results are loaded in the DQT, they can be downloaded as CSV or file attachment, and now export to CBrain using its HTTP API. So now it's time to go to the first exercise. The goal of this session is to get you to run a CBrain task using a selected set of files in the DQT. So first, I'll do the exercise and leave you some time to complete it. So I'll go to the UK Biobank Loris instance, username, password, I'll log in. And this is the landing page. This is right into the DQT, the data query tool. There's a various section there, and I'll ask you to go to load existing queries and load the admin tutorial 1 query. A success message should be displayed. And I'll briefly show you the query. It contains only two fields, the year of birth and sex of participant, which have year of birth smaller than 1939. And then once the query is loaded, you can run it, and you should expect 582 rows. So I'll go back to the presentation and wait for green check marks.
Yeah, and this is just a good point to mention again. So those who are signed up here that would be able to log into the UK Biobank LORIS instance are those who are part of the application, right? Who are part of the agreement for, so not necessarily everybody who's here, I know. So if you've been confirmed, and you should have received information about your LORIS account specifically for those who are part of it and be able to do it interactively.
But for those who are not, there's still a great value in
exactly
how quickly you can select and filter data with a few clicks on the web without needing, elaborate programming or statistical skills. And that hopefully will trigger, thoughts in terms of, your own infrastructure, or what you'd like to do. So Xavier, back to you.
Yes.
Money aside for retirement, and that's okay. You're going to have other priorities, because then maybe once the kids are in school, you don't have daycare, childcare you got to pay for.
I think I hear somebody in the background.
Oh, can I just ask a question? Sorry. If we were already part of the group application to begin with, do we need to fill out the rest of the form?
Yes, you need to have filled the request account page from the LORIS instance.
Yeah, so all participants, so there's the twofold. You might have been part of the original application, but then there's other additional information. So it has have to go through there. If you find off the documentation page for NeuroHub, there is a separate LORIS form you have to fill out, yes.
Okay.
It's on the main LORIS page when you log in.
So I'm not seeing any green check marks. Maybe this workshop is not so interactive.
For this phase, yes.
So I'll move on, and if people have questions, then just raise hand, please. So the second exercise is to go back to define field and add the T1 images. So I have my previous result there. I'll add more field to the query. I can type T1, and you might notice the suffix there. It concerns the instances in the UK Biobank, T0 being a dummy instance in which derived data are added. But for this example, let's just add the nifty image. It should be the second field right there. You can see that it's a downloadable field. And I'll just select the one from the imaging session. I'll stop briefly. The field, they are all prefixed with an ID. They are effectively the field ID that you can find in the data showcase. So if I take the 2252, you can see that they are, this is the T1 structural image, the nifty file. There's a lot of information there. It explains that it's labeled using the instancing 2, and they run from instance 2 and 3. So instance 2, it's the imaging visit, and the second one is the repeat imaging visit. In the visit list there's the imaging visit and the repeat one. I only select the first one for the export part. I will go back to run query, and we should see a double amount of rows. If I go to the longitudinal view, then all the images are loaded along with the previous fields. At this step, any file can be downloaded directly in the browser. This is effectively querying or making reading the file on Beluga without keeping a copy. The same thing for the images. So instead of waiting for green check marks, I'll go to the exercise number 3. So in this exercise, we are adding a filter. To do that, we are adding a rule. I'll add a filter for the COVID positive people that fits the previous query. So I go to define field. And in this tab, we can add a bunch of groups of condition or a single rule. So just to reduce the number of rows, I'll go and select the COVID-19 instrument, in which there's a result data field, and I'm selecting the operator equal for 1. This is true. And then go back to run query. So there's no new field, just filter down the number of displayed row. So I'll continue. I don't see any raised hand.
Well, maybe let's just see verbally from people. Are people able to, maybe they can type into the chat, have they been able to go through login A or exercise 1 into the Loris UKB instance? And then secondly, conducting the second exercise of running the first similar query and then adding the data field. Can people maybe just give us some remarks in the chat just to let us know?
Yeah, so if you're having trouble logging in or something like that, some of the chat remarks are suggesting that you have access to, a different instance, and that's fine, but we can get your account hooked up pretty quickly here. So if anyone is having issues, let us know.
Or even if they have success, then let us know as well.
Mm-hmm. It's good for you to be able to play with this yourselves, right? So what you're seeing here is a very specific set of query, like a query that was already pre-constructed that was nice and simple, but going through the motions will help you understand how to do this for more elaborate queries in the future.
Yeah. Just to answer, I think it was Etienne made a remark. There should be, at least based upon registrations, 11 participants. I don't know if they've all appeared. I can't see it in the full width, height of my screen. There should be 11 participants who have all their accesses appropriately configured for logging into the Loris instance, but also for the subsequent exercises.
Sorry.
And if maybe Xavier, I think you showed in your window the number of returned entries, for example, to display. For the particular query that you example, if people wanted to compare their result, their kind of output to see if they have the same return number of entries as you do. Just to
I can go back to the
yeah, we could do it again one more time just quickly.
I think so, yeah.
And I'll answer a question the same time. I'll reload the module and go to existing query. I'm loading this query. The question was, I'm selecting a field, so any field, and when I run the query, I don't see any result. That's because we've put a guard to make sure that not all the data is queried, because this is really, well, it's not efficient, and we require for at least one filter to be added. I don't remember who asked the question, but this should answer it.
Yeah, so the answer is basically add a filter of some kind, and then you will see that data. There's no point in us having all these different queries run at the same time with all of the UK Biobank data right now. That would be a serious load on the system.
Yeah, so it's clear as the steps outlined in the top where it shows indeed you do load a query, but then you want to define the fields and the filter really to get to the next step.
So you can see Xavier going through it again. He started from the beginning, creating and loading. He's now defining fields. So you can see how he's doing it. He's now defining the filters. He just defined the filters, and then it's about clicking run query.
In this case, there is about just 1,164 entries, it looks like, that were returned from that particular field and filter.
Yeah. And
so, go ahead, Xavier.
If the view is to longitudinal, then it's 582.
So basically, what you're seeing here is the ability to, within a few even seconds, but let's say you do have a very elaborate query, within a couple of minutes, you can define your fields, you can load an existing query to begin with, or you can define your fields, you can define some filters, and then you can get that data. So that's the point where we would like everyone to be right now. You can play with it a little bit now, or even later when we get into the more of the workshop aspects of this meeting today. But right now we're more in the demo side of it. But we just wanted to give everyone the opportunity to catch up to this point. So glad to see that one of those queries worked. And are there others that are having issues, or people still stuck on the account, getting account access or anything like that? Feel free to pipe up. Otherwise, we will continue on. We'll give everyone a few seconds.
Yeah, maybe while we give it a little extra, I think there was a question from Fabrizio about there's a field in UKB that I can't see in the LORS. Is it correct? The field is the death register 0. I don't know if anyone wants to field that query now, or Xavier maybe.
I can answer. It's effectively one of the upcoming feature. We are planning to add fields to the not only the DQT but also the UK Biobank data set that is on Beluga. Not every field were available when we imported it in LORIS, and more fields are requested. Not every day, but more field will be requested.
So that's one of the ones that was not in the original download then. Okay.
Okay. We're getting a request for an account being created, and somehow it's not, but we'll try to work that out.
Are there any other people who have, like I said, I can't see exactly, there should have been 11 people configured, or is anyone other than Zara able to achieve and succeed? Are there people who have not been able to? Oh, I see him on, thank you. Yeah, I think in your case, Catherine, is because you're not part of, you're one of those particular groups of people that right now are not part of the agreement process. You would just need to go through the documented process to request access. So in your particular case, that's why
it sounds like a few pending accounts that maybe Xavier.
Yeah, and just to clarify about the pending accounts, it is more than just also configuring accounts on the lower side. I think as most people should have seen, if they haven't yet applied, the first step for all of this is applying to the UK Biobank. And as we think we noted on our documentation page, there's an administrative process behind it that, even in my particular case, that took them 2 calendar weeks to proceed with that first approval. So that's the first stage, right? So beyond that first stage, then once it comes back to us and you complete the secondary step of registering through the LORIS online web form, that again is a secondary step. So those are 2 mandatory steps that we have to go through to make sure that we adhere to the criteria to be applied for managing the access to the data. Same thing, and even increased, even if you were part of the original application, that secondary step has to be completed as well. So I hope that clarifies for people who may not have been communicated that they have a LORIS account. This was mentioned in the, I think in some of the registration information, but we understand that it takes time in getting the back and forth completed with the UK Biobank and the AMS system. Like I said, that can take we've seen up to 2 weeks. And then accordingly, so that's why we give a little bit of notice, but we understand in some cases it may not have been sorted out in time for the workshop today.
Okay, so I think probably we should move on. We've gone through, a couple of quick passes on what building a query looks like. Like I said, that you can build much more elaborate queries, which we will, I think, have time to do later. So for now, why don't we we can go to the next step and, show what the different dissemination possibilities are and go from there.
Yes, okay, thank you. Basically, in the DQT, there's the download as CSV button, which download everything there. You can also download all the files. That's one of the reasons why I've asked to add a filter, because downloading a few hundred files takes time, and I would like this to be successful for everyone during this presentation. So I'll go back here, run the query. Now back to file. I'll download the thing, small progress bar, and then the download. You can notice that the structure here is the same that you will find in the Beluga dataset, in the dataset on Beluga. It's a bits dataset with every participant having session and for the T1 in the anatomical directory. I don't have an Nifty viewer, but you can see the JSON file. So without having to connect to anything else, you can have an access to T1, but also the other modalities directly through the DQT.
Just to mention, there are some viewers, not for the JSON, but there's viewers within the CBrain side of the NeuroHub, right? So
yes.
So once people open files there, then we have an embedded viewer for some of those formats.
And as for the last exercise, it's for the third option of dissemination, we will now export it to NeuroHub. To do that, you go to your NeuroHub My Account page. I might need to log in now. Okay, My Account. And there's 3 buttons at the top. The third one is New API Token. This is generating a token that once used will be attached to your IP address. So for security reason, you can't share it once it's used, but it allows you to use it through another application. So in this command prompt, I'll just paste my token and click export. And with this success message, there's now links to go to the CBrain file viewer, as Brian was mentioning. You can see that the results are available in a CSV file, but also the CBrain API allows to create what is called a CBrain file list. This is providing a list of file name in a specific project, which is the NeuroHub UK BB Access project. This is why you need to have access to that project. It creates a single file that is mapping the file ID in CBrain to the base name. And if you go back to your project, you can browse through your files. I'll find the last created. This is the one, 1420, 14.2. So this is my NeuroHub list. Oh no, I don't have to click, sorry, my mistake. I'll go back, select the right file, and then click launch. There's a list of available installed tool on CBrain. FSL Bet is the one we've select for this workshop because it's running on nifty files and it's fairly quick. You can select the 509 version on Converter 2 and then launch FSL Bet. There's a few mandatory parameters there. I'll go back to the presentation just because I've listed them right there. So I'm at this step, task control. You need to first select where to save the result. So the result will be saved on main store. You select your CBrain file list as input. Any string will do for masked file. And then there's two main program parameters that needs to be set for nifty images, binary mask and fractional intensity threshold. And then I'll launch the task.
So maybe can I put a pause just for a second here? I want to just emphasize the context of what you're seeing here, because you're seeing a lot of very specific details in terms of, you know, transferring data and launching a specific pipeline. If we can just go back to the dissemination part, if you can go back to the DQT again, Xavier, one more time, I just want to reemphasize what is going on here, okay? Because it might be hard to keep up with this in real time. And so probably, you know, seeing it once is good. Seeing it a second time might help. So you have two options at this point. You've played with the query tool, you've selected what data you want, and you can either download it locally on your machine, as Xavier showed you. It downloads right in the browser. It makes sense if your data sets aren't too big. If you're dealing with, say, scalar, you know, more scalar data, not very large, you know, imaging or genetic data sets, then yeah, download it in your browser, create a little comma separated value, like a statistical like input file, and then take it and do whatever you want with it. Some people run local statistics on their machine. They use something like SAS or SPSS or R or whatever software you're accustomed to. Or in the case of something like the UK Biobank, what the second part of what Xavier was showing you with all those API tokens is that you might want to not, you know, copy huge amounts of data, right? It wouldn't make sense that the UK Biobank is, you know, even just the imaging data alone is, I don't know how many, you know, terabytes of data. So you're not going to probably want to download that locally on your machine. What you might want to do is not even duplicate the downloads at all and use where the data is currently being stored under this allocation and running tools in that exact same location. So that's why we have this export to NeuroHub, export results to NeuroHub, because it's not actually transferring any data. So, you know, that's the second part of what Xavier was showing you. And maybe it's worth just going through that one more time before we get to the point of choosing a specific tool, just so everyone is clear on what's involved and what the steps. Maybe we'll take one more minute and go through that quickly again so you understand exactly how that works. Xavier?
Yeah, I can export it again. I go back to this step where I'm prompt for an API token.
And by the way, a little bit of context on this procedure. You know, data transfers can be a real pain. You know, even if you are programmatically inclined, trust me. So there's all kinds of issues with firewalls and file mounts and things like that. So being able to do this by just, you know, taking a token from one system and putting it into another and then saying, yes, export, it might sound cumbersome, but really compared to what you would need to normally do, it can be, it's so much simpler. So you do what he's showing you right now, and your data is basically, the sub cohort that you've chosen is basically selected and already available for you to do whatever processing and analysis you want. So with that, go ahead, Xavier.
Yes, I can export it again. There's two things going on there. First, Loris will wrap all the table there in CSV file and send it to CBrain so it can be saved in your user project. And the second one, it's extracting the base name that are known by both Loris and CBrain or NeuroHub to create the CBrain file list.
Xavier, one question. They're asking if you can show where you get the token from again. So the
Yes.
token you get from the NeuroHub side, and you can just show that directly.
I'll log out again.
So you log into the NeuroHub side of this, because right now we're in the Loris side. So you go to the NeuroHub portal where you would do all the processing and computing aspects. And Xavier?
Tab there, the My Account page. There's 3 buttons at the top. And there's one for a new API token. And once you have that string, you simply copy it and paste it.
So you copy that, put it into the Loris data querying tool. And then
So
we'll transfer your data.
I'll just open a new tab to show that a new file has been created as of 2 minutes ago. Well, 2 new files, the CSV one. And the CBrain file list.
So that solves the data transfer issues, right? A couple of clicks, copying and pasting a token from one platform to another, and your data is whatever data you've very elaborately chosen is now available where you're seeing it within this CBrain interface there. And now you can do things to that data. You can process or convert or whatever it is you want, and you can choose from any number of pipelines and tools that exist there. And this is the part where Xavier will take you through in more detail.
Once this ID is found for a file, it means that it's known by CBrain. I was not intending of doing a full demonstration of CBrain, but I think I can simply say that once a registered file in CBrain is known, it can be copied over all the HPC network that CBrain is attached to process any tool that is installed there. The other thing that I was not planning to show, but it's a nice side for people that have attended the previous workshops. Everyone should have a NeuroHub project in which once the file exists in CBrain, they should be able to copy the file to their storage. If they've attached a storage on any host that they have access to, it can be your Beluga project space, or it can be your home Linux server. If you have a storage known by CBrain, CBrain can move this file, but also the results of the task that have been launched. I'll go back to this task. It's completed now. What we can see there is that it's the batch view of the CBrain interface. With the CBrain file list, CBrain launched 2 distinct tasks on Converter 2. And each of the tasks, you can see the detail of the task, provided parameter, input file, but also the output files are there, as well as all the processing log, what happened with within CBrain, but also on the selected HPs.
So just a quick high-level recap of what is going on here is now that the data is transferred, a specific tool, FSL-BET, which basically does brain extractions for fMRI data, has been added to, has been run. Sometimes these pipelines take a long time to run. If you're running this on 40,000 subjects, that can take weeks potentially, or at least days, depending on how much compute resourcing you put to it. But we just did this for a couple of subjects and using one very specific tool. And so there are a lot of details here about the provenance of the tools, logs of what's being run, parameters that you would want to choose. There's a whole world unto itself just on this side as well. So you being able to run a tool through a web portal is also simplifies the work a lot of times. Many tools are command line and not so intuitive to those that are not, computer literate. And so this helps in that process. If you're an expert in, imaging, but maybe still need a little bit of work on the computing side, you can probably benefit by, looking at this from a web browser and choosing the parameters you want to run your tool on, and that's all here. So that's what Xavi is kind of showing, just in case this is going a little fast for anyone. And by the way, is this going okay for people? Maybe raise your hand if you think this is going too fast. Any hands? Raise your hand if this is going too slow. Okay, I won't ask if it's going just right, but I'm going to assume that that's the case. So anyway, that's the context that I think. Yeah. And
then really the benefit there is, as you talked about, is getting that in situ processing essentially done in this particular case of the UK Biobank. You're not worrying about, as you said, the complexities of data transfer. You're not worrying about the duplication of the data. You're taking the original source data that we provide through the UK Biobank. You identified a filtered down selection of that, and that is then transparently passed along as input through a graphical user interface way for launching the tasks on resources that you would otherwise have a significant, needless to say, barriers in terms of, as was talked about, in terms of the tool preparation on the systems, trying to compile the tool, let alone executing it in a reproducible fashion. And then you have to go back and say, okay, how did I run that tool? Or how did my colleague run that tool? This way you can actually see exactly the parameters that were fed to it, and it's rerunnable, it's reproducible. So that information is all available there.
While you're speaking, I'm just showing the CBrain viewer for the result.
So this was just one tool even. So there's a variety of other tools. And even to mention about, Samir, you talked about the processing that goes on. This was a quick example. So I think I mentioned earlier on, even though we have, the main data set taken from the UK Biobank, we've been running already a significant amount of pre-processing on that data so that we would then produce derived outputs and then making those available back to everybody so that we avoid necessarily needing people to rerun the various different tools. So that's going to be made available back into the system as well.
Okay, so I think that's sort of, where are we at now? So does anyone want to see the pipeline be run again? Is anyone unclear about that part, or does that make sense? Feel free to speak up. There's no harm in asking, to be clear. one thing, Uku is having a bit of a problem with the token, but he's asking if different email accounts for different platforms might be a cause. It might be. So maybe one of the helpers can address that while we continue on. Also, another question. The query does not show the image link. Is this problem my counter or the process? Good question.
Well, I think the image link, are you referring, because there is both the cross-sectional view, right, that Xavier you're showing, and the longitudinal, and the cross-sectional didn't show the actual image, right?
Yeah,
Or I was wrong, sorry.
Down
Further below, okay.
and either add more result or switch pages.
Okay, so hopefully those are slowly being addressed. Any other questions about the process? I mean, the full context here is you're going to be querying data, you're going to be transferring data, you're going to be running data on an HPC using a specific tool of your choice. And there's probably more details about that we could say, but that's the overall idea of what we're trying to do here. Another question, maybe it's a good time just to address them right now. Fabricio is saying, my Loris does not have a fourth column with the link, with the files link. Is that, have you clicked on cross-sectional versus longitudinal in terms of the views?
No, as Xavier is showing right now, it's probably because he didn't modify the query to add the last column.
Ah, okay.
Make sure that the T1 structural.
Yeah, maybe walk through again that, Xavier, just to visually show it.
One last time. It's easier.
So again, it's loading the existing query, the tutorial one that's specifically there, right? And then defining the fields.
Finding the field means adding columns to the data query tool results. So make sure you select the NFT image. The session selection is mostly for efficiency in the query. There won't be more or less result if you select all.
Adding a filter of some kind.
Adding a filter, I've chosen the COVID one, just because, well, we did not know how many people will run the same set of tasks in parallel. So adding a filter there, running the query. The default view with cross-sectional, there should be 4 columns.
Does this, hopefully this is more clear? Any other questions? Another question, will you cover how we can access the data saved to NeuroHub by making an SSH connection to Graham? I'd like to access the data through the terminal so I can run custom Python code on them using Graham's resources. Right, so we can maybe talk a little bit more about how you would run very specific code. Should I dive into that, or maybe?
I think this part was covered in the notebook workshop.
Yeah, we gave a notebook workshop specifically that showed about accessing some file, but wasn't UK Biobank file specifically. So that typically works within because we have a singularity containers that run for that.
And it was not over SSH, it was the HTTP one.
Right, but people can still, so if you look at I can point the link to it, but if you go through the NeuroHub website, and under documentation, there's section 5.1, or section 5 specifically. There's 5.1 and 5.2. They give instructions there about how to access the data from the Compute Canada resources. Because these files are resident specifically on Beluga because of their volume, you would have to run your code, specifically your Python code on them using on the Beluga resources at the moment, because that's where the data files, the squash FS files are located. So that section 5.1 and 5.2, thanks, Xavier, provide very detailed instructions on how to actually mount the squash FS file and work with it.
Now, it is possible if you have your own tools to, add it to sort of the CBrain environment, containerize it and, describe it. And that maybe we won't get into the full details of that right now, but there's a whole other training session around things like that we could talk about. So for now,
and I encourage people to, if there are things that you're not seeing today that you'd like to learn more about, either as follow-up to what we discuss, there's the survey, which we'll share the link with at the end. Please feel free to put it in there. So that way we can get a good representation of what people are also looking as follow-up or other sessions that we can provide.
Okay, so
and I think there was a comment about the genetics. I assume that modality is yet to be available in Loris. I believe that's true, right?
Yeah, it should follow the same path, meaning mounting the file using SSHFS and Singularity to have them downloadable or referenceable through NeuroHub.
So we haven't yet included it because we just recently downloaded all of it. It actually takes weeks to download some of this data. That's how big it is.
Yeah, just recently finished. So that's just the mains first download. Then as was mentioned, has to be incorporated into the structure of files and then pulled into Loris and CBrain side of things. So that'll be coming. And then when it's in there, the 5.1 documentation page will be updated to indicate that it's available. So you can see in the tables right now, some of it's marked available, some of it's marked unavailable. As soon as it is available, it'll be updated accordingly on that page.
Okay, so where should we go from here then? I'll pass it back to Xavier.
Okay, I'll move on with the upcoming feature. As mentioned earlier, we want to add more data. I'll go reverse. More data, more fields, more modalities. There's also the data field and encoding that requires users to navigate between the UK Biobank data showcase and the data query tool. Having a helper there that describes both the data field list, the encoding for each data field will facilitate the use of the data query tool. There's features available within the CBrain API to store file name in specific project with specific file name. This is yet to be implemented. While creating the CBrain file list, it's referencing files that are in BIDS format. So having a file list that contains a BIDS directory is another feature that I think will be useful for any Python or PyBids user. And of course, the user's feedback that attendees and future user will provide will be answered and gladly integrated in the data query tool for the UK Biobank imaging instance. You can find documentation, the page I have briefly showed how to access data, but a whole bunch of documentation of how we've added the UK Biobank and more generally the NeuroHub platform documentation can be found on GitHub. Any question or problem can be addressed to the support at neurohub.ca. And I think I'll stop here. Thanks to the NeuroHub team.
Thanks, Xavier. If maybe I can get you to scroll to the next slide. I think that should be there afterwards.
Oh, okay. There's
Before
or go ahead.
Yeah, I was just going to say, I know there's another question that just came in the community, in the Zoom chat window, but I'll cut and paste it into the chat to everybody. But I would just ask that those of you who have attended, if you could please take a few minutes. It's a very short survey just to get your feedback, like I said, about the workshop today, but also, like I said, about if there's other information that you'd like to see spoken about and workshop subtopics, whether it's just talk about how to use it to access data from the command line, how to make use of importing your own data, connection points in for different data transfers, other more specifics about the DQT in general as it can be applied to maybe other data sets. Please let us know via that feedback in that particular survey there, which I've just now cut and pasted into the chat window.
I was going to say that'd be good to put in the chat so that people can click on it.
Yeah, exactly. Since people, exactly, I was cutting and pasting at the same time. I see one question from Ugu. Are we allowed to download UKB tabular data to our local machines? You're allowed to, but doesn't mean you want to.
It's just you have to be careful with the those data are only available and usable by people who have actually signed the UKB transfer agreement, like the data usage agreement. So I think that's so. You have to be careful with both the safety, the security aspect of those files, and but you are allowed.
Yeah. So even if you download it to your local machine at your lab, just because you yourself may be entitled doesn't mean the other people, as JB said, every person who's going to be accessing data has to be authorized both by the UK Biobank and by us as the secondary managers of that data set copy that we have making available through to the McGill community for that. So there's an obligation on all fronts to do this proper tracking and authorization of access, let alone the practical elements as well in terms of having other copies and then managing and having to understand this provides linkages all the way through from your intent process. Okay. Are there any other questions or comments from people that they want to mention now? Verbally, we can stick around and chat with people. They can either post them in the text window or we can verbally communicate as well if that's easier for anything that's more, I would say, more detailed or more specific to your particular interest for this particular topic. We're here to help and discuss further with you, whether it's on the DQT side or the processing. We're happy to field your questions. See one more. Is there a way to see a variable values when filtering? Right now, the filters have to be guessed.
I'm not sure. So what do you mean, Uku, about the?
Or looked up online. So you mean like, I guess in terms of what, if we say select a certain value is equal to 0, what does that 0 equal actually represent, I believe?
Yes, I think. No, there's no way within the data query tool. You need to navigate between both the UK Biobank data showcase to have a list of encoding. That's one of the features that was mentioned, to have a helper that will both describe give more information about the fields and the actual encoding used.
So one comment, we do have a feature called data dictionary module in Loris, typically for most projects, where you can exactly see that. Question for you, Xavier, is this something that we might want to enable, possibly?
It's already enabled. We're using it to describe the data type.
But can we then show more granular details somehow in there? Like what is the stumbling block between getting all the information versus what we have for the UK Biobank?
It's that the encoding is shared amongst the data field. So many data fields can use the 0 and 1 to describe the value false and true. There might be something to do with the common data element that we would want to use to describe the categories that we've created, the instruments. We might need to go there, but we could also store the encoding table and reference it for each field.
So future work potentially, but for now you have to manually go to the UK Biobank site to look up what exactly the details of your variables, and then our role here is to make it easier to query, to transfer, and to process. That's our main agenda right now. But there's obviously more that we can do.
Yes, we're not further or behind what's available on the tabular data in Beluga. We're displaying what we've downloaded.
Yeah, so there's consistency in that sense from what's visible, right? So it's coming from the same source. There's no separate copy that people would see otherwise on Beluga. Whereas though there might be some elements that are on Beluga now that may not just be visible through the DQT part, right? I think there was a question, how to access the data from the fields not already in Loris. I'm not quite sure. There's the data from the field. So if it's not visible in Loris, but might be visible, the only alternative mechanism right now that we provide is if they access it through the command line otherwise.
Yes.
Right. Or the NeuroHub portal, but they'd have to navigate to find that. Is that correct?
That would be through the tabular file on Beluga.
Yeah.
Another question, is there a way to generate a list of links to the Beluga files from the query in Loris to run, to afterwards run analysis directly on Beluga?
I was going to reply to this in the chat right now, but I might as well answer out loud, I think, right?
Yeah, sure.
Sure, go ahead, Pierre.
Basically, what I was typing is that the connection between Loris and NeuroHub CBrain is made right now by base names. So you select a bunch of files as registered in the Loris side, and the list of base names, just the plain base name without the full path, are sent to CBrain, and then CBrain makes a mapping to what it knows about for such base names found in a particular project, in this case, the NeuroHub UK Access project. And given now that these files on the CBrain side and on the Loris side, they're stored in a very abstract way on Beluga in a SquashFS file system, which is mounted through a container. This means that there's a whole lot of path translation, very complex path translation that is happening on the CBrain side in order to find the files. But yes, there would be a way to figure out, what are the final paths of these files on Beluga from the CBrain side. It would require a little bit of a coding, and maybe we could program a new utility to generate such a list for you as a user if you are interested in doing this, because within CBrain, within the NeuroHub, this information exists definitely. So for you with a power user who wants to directly access the files on Beluga, then just contact us and we can deploy or implement this functionality.
But can I just make sure I understand the use case? Because you do have access to the files on Beluga directly through the SquashFS and Singularity container, you can actually have access to those files.
True,
but they may not have filtered them. They've made, right?
Yeah, exactly. So on Loris, they've applied a set of filters, and they've now created a list of 243 particular files that they want. Now they want to match this to, yes, they have access to all the files on Beluga, but these 243 specifically, how do you get them out of Loris and into some kind of script that you want to run on Beluga?
Yeah, I understand. That's a very good question, actually. Something that would be useful for sure.
If I can clarify,
Go ahead, please.
Can you hear me?
Yeah, we can hear you.
So yeah, the use case is more, I'm a computer scientist, so I think that the question is more like, is there a way to just get the query first and have a list of subjects, which is the case, and afterward maybe a way to map to their folder on Beluga. So maybe it will include some like import and SquashFS stuff in the header of the file afterward to get that, but it's more to get the list as like a text file or something to more easily manipulate it afterward in the code, or move them, or not need to duplicate or copy them locally.
Yeah, but given that Loris already has an export feature where you can export a CSV, then if your CSV has those base names in one of the column there, it's pretty easy at this point to take the CSV, bring it to any supercomputer cluster, and now just replace those base names by full path names, because it will be easy to, given a base name, to figure out, okay, which subject and which session they're part of. The naming conventions for BIDS data is very clear that you repeat the information in the base name that allows you to recreate the path.
Thank you.
Okay, so I'm trying to see if I missed any questions in the chat. I don't think there is. There any? I don't think we've. I think we've covered it. Is there any other questions that we may have missed from anybody that through? I know there's been lots of communication back and forth in the chat and elsewhere, but. Going once, going twice, everyone. So I'll remind people take a look again when we put it in the final chat window. If there's, again, the survey, please take a look. And if you have any suggestions or other questions, please don't hesitate to either put it in the survey, or you can email us at support@neurohub.ca, and that goes to the team here. Sorry, go ahead, Samir.
Yeah, so maybe just to give one more final piece of context for this whole process, right? When I first started doing any of this neuroimaging kind of stuff, it was very manual. It's still very manual in many realms. I see people that, get a bunch of data, they create several scripts to parse through that data. They then have to, if it's large amounts of data, they have to transfer that data. That first step already can take a while. If you're so computer inclined, then, maybe it's quicker for you. If you are a researcher who, doesn't have the time or the inclination to go through and filter through all this stuff, then if you're lucky enough to have a programmer down the hall, maybe that'll happen on the quicker side. But now you've got this, sub cohort of data that you want, which now you can, with a few clicks on the UK Biobank Loris instance, you can see how you can filter through whatever you're interested in. Then the data transfer issues are, they're abundant. I am a computer programmer, and I still have trouble moving data from one server to another a lot of times. It's not necessarily always a technical issue. There are, barriers in terms of firewalls or VPNs that you have to go through. And so this can very quickly become a very problematic case. So step two, we simplify that process tremendously by just not needing you to worry about any of those kind of things. So that's the API token side. And the third part, which is the processing, in itself can be a beast. Not everyone is an expert computer programmer in high performance computing environments. And so there are all sorts of details that both the CBrain core platform abstract for you. So you don't have to deal with, batching queues and launching jobs, as well as what you've seen in terms of being able to take the data that you have chosen and run a pipeline, a nicely described and organized pipeline. There's a whole aspect of data sharing that also benefits you, even if you are a expert, processor, so to speak. You can collaborate very easily with other people by saying, look, I've selected this data. I want to run this tool that you've created. You can share the results, you can share the tool, you can do all these things in a very easy way through the web. And all you have to do is become familiar with a few of the interfaces that we've shown you today. So that's the goal here. And if you have any questions about any of them, because there is still a learning curve, a little bit of a learning curve on familiarizing yourself with some of the interfaces, then we're here to, we're here till 4 o'clock to answer any questions that you might have. We can go through any one of these features in detail, and we've dumbed it down today to the specifics of transferring UK Biobank data, but there's a whole other world that we can talk about as well. So I just felt it was worth adding that context one more time.
Well, thanks, Mayor. And just to mention, I've shared, and I know everyone should be able to see it, I hope, is just that this is a snapshot as of the end of December, the tools that are already pre-built and accessible to users that, don't have to be compiled, don't have to be, the parameters, there's certain defaults set. People can basically then have these loop and arrays over the various input data samples. So there's a whole slew of these, and these are all, as I said, pre-defined within the interface.
And just to add to what you were saying, Samir and Brian, one of the key things for the UK Biobank is really to be able to share the derived data, which takes so much time to be processed. So I think that's one of the very strong advantage of that project here, that you would be able to benefit from processed data by the team or by others.
Absolutely.
And I need to sign off. I just wanted to, before signing off, to thank you, Xavier, Samir, and all the team, and Brian, because I think that was a truly excellent training session. So I enjoyed that very much myself.
Thank you everyone for listening.
Take care.
So for those of you that would like to stay on and continue with the detailed workshop aspects, we're here to help. We'll stay on the call for a while longer.
Yeah.
And for those that were just here for the demo aspects, feel free to stay or go. But thank you.
Yes, thank you everyone for taking your time to come see this. And like I said, please do fill out the survey, and we welcome your suggestions.
The survey is of particular importance because you have a great deal of power. Whatever you say in that survey will definitely be internalized. I don't know if all surveys do that, but this one, you have my word. So anyone with specific questions? I know there were a couple at the end there that I don't know if can people actually speak right now, Brian?
Everybody should be allowed to turn on their microphones to speak. Yes.
Maybe it'd be easier if people just.
Yeah, I see a question from Ted. I'd be curious, more examples with some of those functions you showed. I guess the tools I've presumably seen. Okay. And then Uku is saying I could copy, and I think that was in response to something that Xavier replied with. Amir, just a quick question. Is AFNI something that'll be added to this list? Maybe that'll be a question more. Is Natasha on the line? I don't know if that one's already in our to-do list or not.
I haven't seen it in my to-do list, but
yeah,
we can look at it for sure.
Okay, so we'll make note of that. Thank you, Amir.
Should we?
And Catherine as well.
Should we try going through these one by one then?
Yeah, I've just been going through. I might have missed some there. So there was the two thumbs up from Amir and Catherine with regard to AFNI integration. And then Ted was someone I'd be curious to see more examples with those functions. Is it just maybe if Ted could clarify, just that I'm thinking about the same thing.
I'm sorry. Yeah, you showed that list of those different tools, and
yeah,
I guess I'd be curious to see, a sort of a researcher going through and running stuff and sort of reporting on those results, and how you would then loop back and sort of adjust those to do the kind of scientific, investigation.
So we're the part of the workflow in terms of that iterative process where, okay, the data is being accessed, you run through it, and then you might want to iterate. And
yeah, or if there's if there's any scientists who are on the session who have used that in an actual study and kind of going through those examples, or if in that kind of example where we can because I'm not a neuroscientist, so it's fun to see it sort of being done in action.
Yeah, one thing, even though it's maybe not seeing it in action per se, I'll let anyone speak up, but certainly I can add the link to it. Just in the last year, we've had about, I think there's about 30 publications that came out that cite using this CBrain task orchestration. So for a variety of these tools, I don't remember them offhand, but I'd have to find the link, but to give examples of where it was used. So that's just from 2020 alone, about 30 papers citing the platform for using it. So I can't see necessarily everyone here, but if there's others on the call here from, I would say, a practitioner point of view that are still around. But yeah, we can certainly take that as an example, though, as a future workshop where we say, okay, let's go through that end-to-end process. You know, typically in the prior workshops, if you look in our website where we have recordings of the fundamentals one, we kind of restricted it to particular tasks. A couple, like one of them was, I think, Civet, and the other one in this case, which we use the FSL bet. But there's other ones that we can go through if there's particular, I would say, ones of interest to people that you know. For example, we can highlight some of those to kind of show the diversity of some of those tools, and then in terms of how they're being worked with and handled.
Thank you.
Yeah, thank you, Pierre, for letting those been processed in Civet. Fabrizio, how do I make a list of subjects in the field death register 0? This field is not found. I think this was comment I was already made. That's one that's not yet in the data that we have downloaded. I think that was correct, right, Xavier? The death register?
Correct.
But we'll update them once we have that in the next update. We'll make sure it's published on the website to make sure it's there. I just posted a link so that people can see some of the what are called the research outcomes. So that's just ones that have come out in 2020 where that have used the platform for processing. And then depending on the nature of the study, they talk about the specific tools in there that have been run as well. Oh, thanks, Natasha. There's another tool that was mentioned, ANTS, I think that was there. Okay.
I have maybe a really specific question.
Yeah, sure, go ahead.
Is there already I think there's already note for ANTS registration. I wanted to know if there's already registration to an atlas already done for this whole data set. I know, for example, Civet used a registration in the pipeline, but is there another one kind of more accessible or something like that?
Does someone want to comment further on that one?
Can you just repeat the question? Sorry about that. I know that we already have an ANTS version of ANTS registration. I'm not sure it's available to everybody because it was a prototype. So yeah, we should discuss about this one if somebody wants to have it.
Because for me, I'm supposed to help Adam for the diffusion MRI processing. And my question is very specific to do kind of automatic analysis. It would be nice to just have a group registration of all subjects to do other analysis.
Okay, and you want to use ANTS for that?
I would say I just want to have a space where every subject is aligned in some case, like maybe not perfectly. I'm more familiar with ANTS co-registration, but co-registering for like thousands of subjects is not the same as hundreds of subjects. So.
Okay, so if I get it, because I am not a neuroscientist, I'm a developer, but you can use ANTS to do that. If we implement ANTS, you will be able to do that via ANTS registration, right?
I think it depends, because by default, ANTS is to register 2 images. So in the diffusion MRI pipeline, the diffusion image, there is an ANTS registration to its T1 image. So now the T1 and the diffusion image are aligned. But I would like an alignment for all T1 image of all different subjects. And I don't think it's possible to do that with all subject in UK Biobank, because it would, like, the ENS co-registration to register multiple subjects at the same time is not made to have that many subjects. Or maybe I'm wrong. But the other way would be to align every subject to a template or an atlas, like the MNI template. So it's just to know if that's already been done. I know in some process like Civet, there's already an alignment with some MNI template, but it's not really accessible.
I have no idea, Etienne. Yeah, I'm not sure.
Maybe this is the question we can take offline with somebody as a source, within the Civet space to get some clarity about that, maybe. Just to get some others. Any other comments? Samir or others who want to tackle that one?
Sorry, I missed the question, actually. I got distracted.
It was about more with maybe Etienne, if you could repeat, sorry.
Yeah, no problem. I will repeat it quickly. So the idea for some automatic analysis, I would like to have all subject aligned in some kind of space. So it could be a T1 aligned space. Normally, I'm more familiar with using ENS registration to register multiple subjects at the same time. But with that amount of subject, I think it might not be possible to co-register all subject into a kind of new template. So I wanted to know if there was some kind of registration to an MNI template already done for all subjects.
I know we're processing the data. I don't think we've done that registration. There actually might be value in doing like specific templates for subgroups because the data is so big, right? So maybe there might be value in, I don't know, having an age specific template or something like that. But that is part of the process data that we will eventually give out. So that is coming. Like they're actually processing it right now and working on it. So what exactly we'll release, that is there's still some questions there, but we'll definitely.
So Samir, just a little clarification. Etienne is working with me on the tractor flow stuff. So his question is actually sort of relates
tied to what we're doing, I guess.
I'd like to say something, Etienne. Given that we've taken all the T1s of all the UK Biobank subjects and we ran them through Civet, part of the Civet pipeline is to register all these anatomical scans to the standard MNI template. Isn't that something that you could use if out of all these Civet outputs you extracted all the registered images? Would that satisfy you?
I think that's possible, but I'm really not familiar with Civet nonlinear transform. And I'm not sure if Civet used nonlinear transform. So that's also the question.
Civet does use nonlinear transformations, and so the details of how that compares to ANTS or any other, processing pipeline, you have to go into the specifics of that, but there's no reason why you couldn't do that.
Yeah,
I mean, so you have the transformation matrices as well, so you can, compare them. So yeah, it's possible.
So in that case, that could be useful. The only trouble I would have with Civet is more than most of the pipeline are and BIDS format are in Nifty, and most of Civet files are in Mink. And if you convert the transform file from Civet, so from what I know, you would need to convert the Nifty image to Mink to apply the Mink tools to register back the image to that space. And I know the Nifty to Mink conversion is not always clear for the space transform.
But we did it. We actually ran Nifty to Mink on all the subjects. We got them run through Civet. So now we can take the Mink files and convert them back to Nifty if you want. It will be exactly the same data, simply in a different encoding.
Yeah, the only thing I'm thinking is to apply the same transform to other files. And I'm not familiar with Mink tool, but if you think it's easy and feasible, then
I don't know
maybe I
if it's
should
easy.
look with you.
I do know that, like Samir said, you do have the transformation files, which would tell you exactly how, how the transformations were made, and that could be maybe useful to you somehow.
So the idea would be to use once civet is processed and available to use the same transform. I think that was one of the thing I was thinking.
Yeah.
Because they use the MNI template. And I think that's the only one that I saw that already included some registration to a standard space.
Yeah, and all the civet ones, we've run civet on all of the images, right, Bear? So
yes,
that's already been done quite a while ago, exactly.
Yeah.
All 40,000 Nifty files were converted to 40,000 Ming files. All 40,000 Ming files were run through civet.
Yeah. I would say the only issue is if we process the diffusion file, each diffusion file will need to also be converted to Ming to apply the same transform, which is kind of a lot of processing for not that good reason. I'm familiar with using registration for surfaces like VTK surfaces and stuff like that. But for T1 image, I might want to look at it, or maybe
maybe
from
it's
civet could help.
Just looking at all of this. And maybe just the transformation files which describe the transformation, you ignore the transformed output in Mink, but you just take the transformation file, and if somehow you can apply it directly to a Nifty file somehow.
That's what I thought at first, but I saw a lot of people trying to do that in the past, and it never works because the alignment of the image and the standard space used for Ming file and Nifty file are not the same.
Right.
So you need to really change the orientation of the transform. And for a affine transform, it's quite easy, but for a nonlinear transform, you have more risk of doing a lot of error. So
right.
I think it's easier to convert and apply it. But that's true that it would be easier, I think, to convert the transform to Nifty and then just apply it, but it's not really possible because ENS doesn't support the same type of transform that civet does or Mink does.
And Etienne, if I'm not wrong, the purpose of all of this is for us to have to build some sort of a level of automated quality control.
So the goal after that is to kind of do some pre-analysis of the data to be sure that every subject was well processed and kind of aligned in a space so we can compare more easily the matrix.
Yeah, the transferring from one file format to another is a bit of a headache. And I agree. There's a whole world of conversation there. But one thing we could maybe do is put you in touch with the people that have done these comparisons a lot. So someone like Claude Lepage or Lindsay Lewis, they've gone back and forth with, comparing pipelines. And so maybe they will be able to make this process a little bit easier for you.
That can be good. I already know a little Claude, so that could also help. I work on some civet analysis, so that is useful. And maybe there's other people doing some processing that required registration that might also that could be included just because they are planning to do to align all subjects in one space. And I think people will use this afterward for analysis because most of the people doing image analysis align them in a certain space. So
you can also talk to Vladimir Fonov. He specifically uses ANTs. He specifically has used the underpinnings of civet, if not civet itself. So he also may be able to kind of guide you through this a little bit.
Okay, thank you.
Thanks.
I mean, this part is very, structural image processing, like is very detailed here, So this is maybe what we're trying to do here is, facilitate the way to, transfer data and process, but this is very specific image processing questions. But happy to help in whatever way we can.
Any other questions? I see there's a few others still on the line. Fabrizio, Laura, Akiko, Uku.
I'm okay. Thank you, Fran.
Okay. Well,
okay, should we wrap it up then? I think.
Sure.
Well, thank you, everyone. And thank you again. I hope everyone can take a look at, like I said, give some feedback, and then we'll hopefully see you at the next workshop opportunity. And thank you for your time today.